for something completely different. So this is a 16th century picture from Lucas Cranach the Elder, and shows the legendary fountain of youth. You've probably seen similar pictures, and in fact, every culture, every civilization has dreamed of eternal life and youth. What this picture shows is that if you bathe at this fountain, you get sort of, you're old, you enter the fountain and you come out young again. You can drink the water and you become young again. And there's people who have spent all their lives, or a big part of their lives, searching for these types of fountain of youth. Ponce de Leon, for example, Alexander the Great. But we know, of course, that such a fountain does not exist. But what I'm going to share with you is an absolutely amazing development in aging research that suggests that maybe there is something to a potential fountain of youth. And this could potentially have a tremendous impact on how we understand aging and age-related diseases. So a small but growing number of research labs across this country, but also in Europe, have found that in a model called parabiosis, tissue can be seemingly rejuvenated. And what that means is that tissues from an old animal can be made younger again by being exposed to young blood. In this model that is called parabiosis, and I'll talk a little bit more about it later, but it's in essence a Siamese twin that we would know in humans, but it's induced surgically. So Thomas Rando here at Stanford was the first to show that stem cells in muscle can be rejuvenated in an old mouse if that mouse is exposed to a young environment in parabiosis. When you get older, the muscle does not regenerate as easily as in a young person. And the same is true in mice, but what he could show is that young blood, a young systemic environment can rejuvenate the activity of stem cells, and the muscle now regenerates like a young one. He also showed similar effects in the livers. Others then showed effects in pancreas, heart, and what we're most excited about is that similar effects may apply to the brain. So a number of labs now, including ours, has shown that the brain of an old mouse that is exposed to a young environment can be seemingly rejuvenated and made younger again. And I repeat, an old brain can be made younger simply by being exposed to young blood in this model called parabiosis. And I'll tell you a little bit more about how we think this might happen and what the concepts are behind it. As we get older, most of the functions, cognitive functions, different aspects of our cognitive functions are preserved well into our 50s or 60s. And you can see here inductive reasoning, spatial orientation, perceptual speed, and so forth. Actually, perceptual speed, video gaming, that goes down very quickly. You can see here the green line. But most other functions, in fact, they keep increasing from 40 to 50 years of age. People actually become smarter for many of these cognitive aspects. But the dramatic thing is, of course, that if you hit 70, 80, or 90, for most people, cognitive function goes down. We know that with age, the connections between nerve cells in the brain, the synapses, degenerate, neurons become damaged, they accumulate all kinds of injuries, and eventually they die, the brain shrinks, and our brains become more susceptible to diseases such as Alzheimer's and other neurodegenerative conditions. The problem with trying to understand why this happens is that the brain is isolated in the head. We can't really get a biopsy. And most of what we understand about the brain is from people who have died. We take their brains and we study it at the molecular level. But all the other things we know about the brain are cognitive. It is cognitive knowledge. So we test people in memory tests, or we can do imaging that looks at structure. And there's a few functional imaging aspects that we now have, where we can look at molecular changes. But most of what we know about the brain is really either from animals, where we can, of course, take the tissue of an animal willingly at any age, or from people who have died. And then we can take the tissue and study how do the molecules change. But that's at the end, when somebody has died from age-related diseases or just from normal aging. So how can we address this? How can we try to understand the brain in a living individual and even follow a person as they age? So one sort of hypothesis is that because the brain is part of an organism and is connected to it, we can study maybe other aspects in the body and thus understand how the brain ages. And what I mean with that is that specifically, if we study the blood, we may learn something about the whole organism. Blood is, of course, the organ that carries cells, for example, oxygen-carrying cells, between different tissues. But it is also the organ that carries information between all the different tissues in our body. And these messengers or hormone-like factors some of them are well understood. And they can carry information from one tissue, such as the muscle, for example, potentially to the brain. So by studying many of these factors, it's almost a language of cellular communication in our body, we may be able to understand something that is going on in the brain and in other organs, and thus learn what happens. So if we look at how these factors change from a young person or a young mouse, a young organism, to an old, we see that the levels of these factors actually change quite dramatically. Our body lives in a very different environment if it comes to the function of, or the levels of these factors. And as I said, many of these factors carry very important information from one tissue to another. So if you just look at these levels, our body really lives in a different environment as we get older. And by and large, factors that are associated with the development of tissue, with growth of tissue, or survival of cells and tissues, they decrease as we get older. And factors that are involved with inflammation or injury, they tend to increase. So there's somehow an imbalance. And by trying to understand that, maybe we can understand part of the aging process. And again, blood we can take from any person at any age. And if there is such a connection between what changes in the blood and what changes in the brain, maybe we can learn something about this interaction. So here's an interesting experiment, a Gedanken experiment, if you will. So we took blood samples from almost 300 individuals that are cognitively healthy. So these were actually seen in memory clinics, and they were all deemed cognitively normal. So they didn't have any diseases, and they had no dementia. We measured roughly 100 of these factors that we know carry information between organs. You might know some of them by name: interferons, for example, insulin, growth hormone. So these are hormone-like proteins that carry information between tissues. And when we measure many of these, as I showed you, because these factors change, we can basically try to understand or measure the relative age of a person and discover a signature of age, if you will. So what we notice is, if we compare the youngest group with the oldest group, roughly a third of these factors, or almost half, significantly change. And I mentioned this before. There's this change in the levels of these factors. Again, suggesting that our body lives in a different environment as we get older. Also, if you use bioinformatics tools, you can try to fish out the ones that most strongly correlate with age, and thus best predict the relative age of an organism. And this is shown here, and I like this experiment very much, although we don't know yet exactly whether this is really predictive, but let me talk you through this. So here, first of all, you have these 300 individuals. Each circle is an individual. You have their absolute age in years here. We call this chronological age. And then we take the top factors that best predict age, and we calculate with the relative levels of these factors, we can now calculate the relative age of a person. And we can call this the biological age. We don't know, is this really true? But this is sort of the information that we can get out of it. And we can ask the question, if this person here, first of all, there's a good correlation overall, as you would predict, because we take the best correlates of aging. and calculate the relative age. So this individual here is roughly 70 years of age, but their biological predicted age is only 45. So is this a person who looks maybe much younger their age, is at reduced risk to develop an age-related disease, and may live a long life? We don't know yet, but this is what we're trying to do. And by contrast, this individual here is not even 40, but is predicted to be 65 years old. Is this a person who looks older, who is maybe at risk for an age-related disease? And if we can understand this, if we can identify such factors, we could enroll this person into a study that is preventive of a degenerative disease. For example, Alzheimer's disease. Maybe this person will develop Alzheimer's much earlier, at age 60, instead of age 70 or 80. So if we can understand these factors, maybe we can treat age-related diseases or enroll people before they have an age-related disease. But all of what I've shown you so far is really correlates, right? We just measure things and then we correlate it with the relative age of a person. So that doesn't tell us whether these factors are really involved in aging and whether they influence aging. So do the factors that go down, do they actually make an organism younger? Do they keep it younger? Or are they just changing for unrelated reasons? So we want to know, are these factors actually modulating the age of a tissue or of an entire organism? And this is where this model parabiosis comes in again. So here what we do is we pair an old mouse that is 18 months of age, which corresponds to about a 65-year-old human, with a 3-month-old mouse corresponding to a 20-year-old person. And again, we connect these surgically, and because this is a wound, blood vessels grow into this wound, and the blood vessels fuse then, and so like in a placenta, for example, you have an exchange of blood. In fact, it's a bit different. There's capillary networks that connect these two animals, and so you have an exchange of blood from the young mouse to the old, and vice versa. And now we can ask, are the factors that we measure in the young mouse versus the old mouse, are these factors actually doing something to the age of that older mouse, or are they just correlates? And in fact, what we see is that these factors can rejuvenate the old mouse. We find that the old mouse makes more stem cells, it activates the stem cells in the brain, and they make more new neurons in the brain. They make more synapses that connect the neurons, so that neurons talk more. There's higher activity, brain activity. And there's also an increased expression of genes that we know are necessary to form memories. So at all levels, it looks like these brains look younger than the brains of a similarly aged mouse. Also, very importantly, I told you with age, inflammation goes up. These mice have less inflammation in their brains than mice, old mice that are paired with other old mice. As we talked about microphones not working.
It turned off or something. Maybe the battery ran out.
So inflammation decreases as well in these old brains.
Hold it closer to your mouth.
So when we then look, so this is all in the parabiosis pair. And we can't really do memory tests in these mice, but you can do memory tests in mice that are injected with young plasma or old plasma. And I'll show you an example in a minute. And when we did this experiment, we could actually show that the memory can be improved in these old mice. So old mice, like people, they have more and more problems remembering things or orienting themselves in an environment, in a spatial environment. And so what we did is we harvested blood from many mice of young age. We got rid of the cells, actually, because we never see cells entering the brains in these pairs. We never see young cells entering the brain of the old mouse. And that, for us, that suggested that it's soluble factors that mediate the effect. So that's why we got rid of the cells, and what is left over is called plasma. And so we injected plasma from young mice into old mice, or as a control, old plasma into old mice. And what we saw is that there is indeed a rejuvenation. We published this a number of years ago, and last year another study that we published, you might have seen in the news, and a lot of people thought it was really funny and scary as well. But I think the potential impact intrigued a lot of people. So since then, we have actually made some progress and asked the question, can we use human blood and get a step closer to testing this idea in humans? And in order to be able to do that, you can't inject human blood into a mouse because their immune system will not tolerate that. But there's mice that are immunodeficient, and they will accept a human graft. So these mice we can treat now with blood from young mice, from young humans, or from older humans, and see, does the blood from a young human also have the capacity to rejuvenate the brain and make these mice smarter? That's what I'm going to show you here. So this is a memory test for a mouse, and it looks probably a bit weird to you, but it's not unlike if you park your car in a big parking lot in a shopping mall, and you go shopping, and you come back a few hours later, and you have to remember where you parked your car. You don't have a chauffeur, and you don't have one of these alarms that just tells you where the car is. So what your brain does, it uses cues in the space, like a storefront or a lamppost, to make a spatial map in your brain, and when you come back, automatically, you know, oh, it was just next to Starbucks. So the mice do a similar thing, and what they have to do here, they're on a table that is about this big. It has lots of holes and a bright light that shines on it. Mice don't like this environment, so they try to escape, but it's too high to jump off. And we give them one hole. All these holes are basically leading to the ground, except one hole has a tube mounted to it, where the mice can escape and they feel comfortable in a dark environment. So they use the spatial cues to remember where that hole is, and this is pretty difficult actually. So if we look now at an old mouse, trying to do this, you can see it's supposed to find this hole here, and it's just walking around and really has no clue where this is. And it was trained for 4 days. 4 times a day, it was trained to find this particular hole. Actually, every day we changed the location of it. But this mouse, and in general, the old mice do not remember this task or have a great difficulty doing it. Now the next mouse is a sibling of it, but it was treated for 3 weeks with plasma from young people. Every other day, it got young plasma. And what you can see is it looks around in the space and then walks right to this hole and escapes. So seemingly this mouse is rejuvenated. It got something in this human plasma that makes it function again like a younger mouse. So what we find is that the old mouse and its brain in particular are not set in stone. They're malleable actually. They can be rejuvenated. They can be made younger in many aspects. Young blood factors can reverse aging, and I didn't show you that, but old blood factors can accelerate aging. And humans have similar factors in their blood. So does that magic work for humans as well? We don't know yet, but we have a clinical study running here at Stanford where we're treating patients with mild to moderate Alzheimer's disease with a pint of plasma from young donors, from 20-year-olds, once a week for 4 weeks. And then we measure their cognitive function. We do some MRI studies where we look at brain function with using imaging tools. And we have sort of daily activities of living that the caregiver will test with the patients. I don't have any results yet, but we're very excited about learning of this. This is a double-blinded controlled study, so we have to wait until the study is finished, and this may take until the end of the year or longer. But I think what these studies suggest is that the fountain of youth, this elusive fountain of youth, may actually not be that far away. It may be within us, but it just dries out as we get old. And if we can turn it back on just a little bit, maybe we can not live forever or be young forever, but we may be able to delay age-related diseases and improve function and health of